Hi, and welcome to the BDNF DNA methylation map, an interactive data visualization platform designed to help you identify DNA methylation sites in the BDNF gene that have been investigated in association with brain-related phenotypes in humans. The application can be navigated using the menu bar to the left. The overview page introduces the application and provides brief instructions for use. The base plot provides a two-scale depiction of the basic BDNF gene structure, as well as all BDNF-associated CpG sites examined in the studies captured by the systematic review through which we curated the data for this application. This plot was created using ggplotly, which means you can zoom in to view a specific region of the gene and hover over elements for more information. Here I've zoomed in to get a closer look at exon 9, and we see an Illumina probe here annotated to HG38 position 276-55578. You can hit the home button to reset the axes. The user-defined plot allows you to interact more fully with the systematic review data. Specifically, we've included several filters in the navigation bar on the left that allow you to customize the plot based on your interest. To demonstrate its functionality, let's walk through a fictitious scenario. So let's say that we're researchers who study BDNF and aging and are selecting CpG sites for a study we are designing. The first filter on the left is for the article reference ID, which corresponds to the tables in our systematic review. We anticipate that most often users will ignore this filter, which is what I'm going to do now. So I'll keep all articles selected. Next, we've included a similar CpG position filter. Again, I'm going to keep all the CpGs selected, but if you have a specific site of interest, this filter can be used. In looking at the phenotype, remember in this fictitious scenario, we're aging researchers. So I'm going to deselect all and reselect the aging phenotype. Note that the plot automatically redraws based on our selection. For tissue type, let's say that in this scenario, we can't feasibly access muscle. So we're going to uncheck this box. For now, we'll keep the color by factor set to significance so we can see both what has been looked at in the literature and of those CpG sites, what has looked potentially interesting. So again, we see the same base plot that we saw before, but this time only CpGs that match our filter criteria appear. Above the CpGs, we see shapes that are studied by CpG by tissue type specific. In this scenario, black shapes indicate studies that were examined but determined not to be statistically significant in association with an aging phenotype, while red shapes were examined and determined to be statistically significant. Just looking broadly, we see that there has been coverage across the spectrum of the gene, but that the significant hits appear to cluster down at this end. Let's take a closer look by zooming in a little bit at one section of this plot. A reminder that this plot is created using ggplotly, so we can hover over shapes for more information. For example, here we see a study by Sato and colleagues in 2020. This study was looked at whole blood DNA methylation and aging, and both the Illumina probe number and HG38 position are annotated there. Zooming back out, let's say we want to remove non-significant CpGs from the plot and color the study-specific dots by tissue. We see that, again, now that we've removed the non-significant CpGs, we see the majority of our results cluster here. And zooming in a little bit, we can see that most of the significant hits have been observed in blood, with one hit here in brain. Zooming back out a little bit, I just want to point out that the shapes located above the gene and the CpG sites indicate CpG-specific site results, while shapes below the gene indicate mean methylation results for many CpG sites. If you prefer to see the data in table form, we've provided a database that corresponds to the systematic review findings and user-defined plot. The table is searchable broadly here in the right-hand corner and more specifically within each column. Finally, if you have any questions or need to submit bug reports, the developer information is on the final tab. Thanks for watching, and we hope you enjoy our application.